Good morning, everyone, and welcome to today's webinar brought to you by the Alberta Canola Producers Commission. Joining us on the line is Dr. Lloyd Dosdall from the University of Alberta to discuss Diamondback Moth, the biology, and the control in Western Canada. I'm going to turn it over to Dr. Dosdall, and he can start his.
Well, good morning, everyone, and welcome to a webinar presentation presented by the Alberta Canola Producers Commission. My name is Lloyd Dosdall, as Rick mentioned, and I work at the University of Alberta. Well, the Alberta Canola Producers Commission is sponsoring this information session on biology and control of this insect because of the situation that's currently arisen throughout much of Alberta and Western Canada. I've recently been sampling quite a number of fields throughout the southern and central parts of Alberta, and diamondback moth populations are really very high in some fields. Not all fields, for sure, but certainly in some fields we have massive infestations. So the purpose of my presentation then is to bring forward some information to help agronomists and any interested listener in making decisions about how to manage this pest. So my presentation then will first begin with some basic information. The life history of this insect in wherever it occurs, actually, the crop damage that it's capable of causing in canola, where diamondback moth come from. I think this is important because it helps explain why we have such a heavy infestation this year in 2010, but for example, in 2008, we had virtually nothing. Cropping effects, what kind of impact farming practices can have on this insect, how to monitor and control it, and finally I'll conclude with some information on natural enemies of diamondback moth because these can factor in quite importantly into control decisions. Sorry about that. Okay, life history. Diamondback moth eggs are laid on canola leaves, on the upper leaf surfaces. Rarely are they laid on the stems. Sometimes they're laid on the lower stems right near the soil surface, but most commonly right on the upper part of the leaf. The eggs are small, yellowish, green in color, and oval in shape. Most of the egg laying occurs at night. Like other moths, this is a night active insect, so egg laying begins usually very soon after the adults emerge. Within about a day, they've mated and are ready. The females are beginning to lay eggs. The eggs hatch quite rapidly in only 4 to 8 days, but this again depends on temperature. Diamondback moth larvae go through 4 developmental stages, or 4 instars. The first instar is a tiny little insect larva that actually mines canola leaves. So it mines the nutritious tissue between the upper and the lower epidermis in canola leaves. So here, for instance, is where a diamondback moth egg was laid. The first instar larva has hatched, and it's bored an opening into the leaf where it feeds between the upper and the lower epidermis. Here is where a diamondback moth first instar had mined canola leaves, and then it had emerged through another opening as a second instar. So the final 3 instars are surface feeders on canola, and the first instar is a miner where it's protected within the leaves from natural enemies, for example. The insects grow fairly rapidly. The larval stage lasts about 2 weeks in this weather, and it can take as much as 3 weeks depending on circumstances. And there are the size of the insect is about a ½ inch in length when it's fully mature. It's possible to confuse diamondbacks sometimes with other green insect larvae that can occur in the field at that time. But there's an easy way to tell. One of the ways is, first of all, the V-shaped structure on the tip of the abdomen of larvae. No other insect larva in canola has that characteristic structure, but also an easy way to tell is to just gently prod the larva from the front end, and if they wriggle backward very quickly, it's a diamondback moth larva. If it tends to coil up or just fall off the plant without any of that wriggling motion, it's most likely a looper larva or perhaps a Bertha armyworm larva or some other species, but not diamondback moth. After it's completed a period of feeding and gone through 4 larval instars, the insect pupates. In this stage, it has now absorbed lots of nutrients from its leaf feeding, and it's undergoing the critical changes that are necessary in order to transform it from a larva into an adult. The pupa constructs a lacy casing around itself. It can be either light in color or greenish, especially when it first pupates, but gradually it takes on a darker color as the wings begin to form within the pupa, and you can see the adult form developing. But this lacy outer covering of the diamondback moth pupa is very characteristic of this insect. Pupation occurs fairly rapidly, 5 to 10 days, again depending on temperature, and then they emerge as a new generation adult. The adults are about a centimeter in length, and they hold their wings in a tent-like formation over the back of the abdomen. And at the tip of the abdomen, there are a series of long hairs that are also fairly characteristic, and they tend to point upwards. But of course, the most characteristic structure of this insect are the scales and the pattern of scales that occur across the back of the insect. And this can only be seen from the top, looking downwards. A pattern of white scales that roughly fuse into a diamond shape or a series of diamond-shaped markings are indicative of diamondback moth. That beautiful pattern of diamond-shaped scales isn't always apparent, though. In this particular diamondback adult that I photographed in a canola field recently, it has obviously been around for several days because its wing pattern is more or less worn off. Its wings are rather tattered at the bottom, and so it can be difficult to identify as an adult, as an actual diamondback moth adult. But these insects mate very soon after emerging, within the first day. They mate and lay their eggs at dusk, with peak egg laying occurring about 2 hours into nightfall. But they will lay some eggs throughout most of the dark period. In the course of her lifetime, which can last about 16 days or so, the female can lay about 160 eggs. On canola, we found in our work that most of the eggs are laid in the first 4 days after emergence, and then after 8 days, very, very few eggs remain to be laid. So then, in terms of its life cycle, it moves from egg to larva to pupa and adult relatively rapidly. And one of the fascinating aspects of this insect that is largely responsible for its pest status is that it just keeps cycling over and over in the season. And what stops its populations from continuing to expand are either attack by parasites or cold temperatures in fall that freeze out and kill the insects. In our area, in Western Canada, they can complete as many as 4 generations per year. And one thing to remember about this insect, because it cycles continuously, we can have all life stages of this insect present in the crop at the same time. And I'm seeing this especially last week in southern Alberta, where adult moths, eggs, pupae, larvae were all present in the same sweep net sample. In terms of crop damage from this insect, it can occur really in all stages of canola plant development, even in the cotyledon stage. It's highly unusual for damage to occur by diamondback in the cotyledon stage, but we've seen it happen. In the outbreaks of 2003 and 2005, we had large infestations very early in the season, and for the first time we had to deal with how to accommodate damage that early in the season. But damage is not as severe when the insect attacks the leaves as when it attacks the pod stage later on. Feeding by the larvae on leaf tissue can have a damaging effect. It can reduce the photosynthetic ability of the plant and ultimately affect canola yield. That is when the situation is very severe, when there are huge infestations. Damage to leaves can be very substantial. But in general, canola can withstand a considerable feeding by larvae on leaves. And it seems that the larvae prefer to feed on leaves. And because the plant is actively growing throughout much of the season, it can tend to outpace the yield loss or any damage caused by these insects by just producing more leaf material without any measurable harmful effects on yield. They can also attack flowers and buds, and we've certainly seen that this year and in previous years. They are indiscriminate in their feeding. They'll climb up on buds, chew openings through the buds. I've seen them very often on flower petals, tucked right inside of flowers. But generally, in terms of bud loss, plants can usually compensate for that feeding by simply producing more buds, unless, of course, the plant is stressed by drought or diamondback moth populations are extremely high. This is a photograph of fairly typical damage that we were seeing a lot of this year and last year, I must say, to buds of canola as a result of diamondback moth feeding, with holes chewed out of the buds, and often if you pull the bud apart, there would be a diamondback moth larva inside feeding on the flower parts. Not always, sometimes they would just chew out the upper portions of the bud, but they were very commonly tucked right inside. The pod stage and feeding on pods can be most destructive in terms of yield. At this time, the plant is putting its resources into seed production. The leaves have senesced from the plants. The diamondback moth larvae need a source of moisture in order to survive, so they abandon senesced leaves, climb up the plant, and begin stripping green material from the stems and feeding on pods. And pods that have been fed upon by diamondback moth larvae rarely ripen properly. The seeds remain green and just simply don't develop like they should. Feeding by larvae on the green pod tissue can prevent the pods from filling, prevents proper ripening, and of course is very destructive to yield. In severe infestations throughout Western Canada, crop losses from this insect can be quite extensive. In the slide that follows that I took in a field near Vermilion in 2005, the farmer had about 100% crop loss from diamondback moth attack. This is a photograph of the field. There was really nothing to harvest anymore by the time that this photograph was taken. But this doesn't need to be the situation. I hope that one thing this presentation helps is to guide farmers and agronomists to be able to make decisions, monitor their crops, and prevent this type of damage. Where did diamondback moth come from? We have found evidence that diamondback moth can overwinter in the prairies. I did, for instance, in the early 90s, have some emergence traps that were on soil at Vigorville, Alberta. The traps were placed out extremely early in the season before frost had gone out of the ground. And we found in several of the traps that diamondback moth populations occurred that could not have come from the outside in, but had to have overwintered on those sites. It was a condition of early, very heavy snowfall that provided an insulating blanket of snow on the ground, and the larvae were apparently able to withstand winter conditions that way. We've since tried a number of attempts to show under what conditions diamondback moth overwinter in the prairies by placing diamondback moth larvae, pupae, under various treatment regimes in cages over winter, and we found it extremely difficult to replicate overwintering. So by and large, it's a safe assumption that most of our populations do not overwinter on the Western Canadian prairies. They arise from airflow from the south. We've been able to show through circumstantial evidence that most of our populations are arising from infestations in southern North America when strong airflow carries moths, which are tiny and easily transported by air currents northward. Also from Mexico, it seems that California can also be a source of airflow north. This was some results from the 2001 diamondback moth outbreak, where we were monitoring through Environment Canada and Agriculture Canada collaborators airflow from south to north. And Environment Canada modeled airflow at 3 different heights and showed that movement of air in early May came from southern Texas, at which time there happened to be a heavy emergence of diamondback moth adults that appeared to be carried northward on strong winds from the south, and as a result billions of moths were deposited throughout Western Canada at that time. Higher air other than surface tended to be carried away from the Canadian prairies. A recent paper by Drs. Hopkinson and Soroka used these models to show much more clearly how effective they can be in predicting diamondback moth populations throughout Western Canada. And the Prairie Pest Monitoring Network is doing this on a yearly basis. So they're watching airflow with the collaboration of Environment Canada and identifying airflow events from south to north that can be responsible for bringing moths to our crops. There are some effects of agricultural practices on diamondback moth, and I'd like to spend some time going through some of those results next. One of the students who worked in my lab here at the University of Alberta did some fascinating studies on the effects of soil fertility on diamondback moth. He had a number of treatment regimes where he had no extra fertility to as much as 5 grams of fertilizer per pod. And we calibrated this so the 1x rate was the recommended rate for application to soil for growing canola, and we went at half the recommended rate, 3 times, and 5 times the recommended rate. We planted canola under those systems, put the pots in cages, and checked the responses of diamondback moth in this situation. And these are some of his results. First of all, in terms of numbers of eggs. It was interesting because diamondback moth laid very few eggs on plants that didn't have good fertility, and they laid very few eggs on plants that had higher than recommended levels of fertility. Most eggs were laid on plants that had either optimal or recommended levels of fertility or slightly below that. So diamondback moth then prefer to lay their eggs on vigorous plants that receive good nutrition. In terms of survival of larvae, we found much the same. Fewer larvae survived at the low and high rates of fertility, and we had generally good survival, best survival, at the intermediate or the recommended rate of fertility. In terms of herbivory, I think this helps explain some of those results. These are photographs of leaves taken from the different fertility regimes, either with no fertility added or a very high level of fertility. And we were seeing quite a difference in the amount of feeding damage to those plants. Most of the feeding damage was occurring to plants that had very low levels of fertility, and we got comparatively less feeding damage when fertility levels were much higher. And I think the reason for this is quite understandable. If you think about why the diamondback moth is feeding on leaf tissue in the first place, it's to sequester the nutrients that it needs in order to develop its tissues, be able to build up the fat reserves it needs for pupation, and construct protein from the nitrogen and so on that are in leaf tissues. And here, a given amount of insects needs to consume much more leaf tissue in order to build up the protein it needs than it does when there's much more nitrogen in the leaf tissue to begin with. So this summarizes some of those previous results where total herbivory, the amount of leaf damage, was much greater when there was very little fertility added compared to when there were much higher levels of fertility. I don't know if many of you who are listening have seen this curve, but the students in my insect course make sure that they know it cold by the time of their exam. But it shows the relationship in crop plants between yield and insect numbers, and it's termed the damage curve. It's a very classic curve in insect pest management. And basically the concept is very simple. When insect numbers are rather low, the plants can tolerate or compensate for a given insect population. Then, as insect numbers increase further, the plant can no longer compensate, and we're starting to see rather substantial reductions in yield. And this is the linear point on the curve where there's a decline in yield with increasing insect numbers. Later on, there is a desensitization and an impunity response by crop plants where even increasing insect numbers further doesn't cause yield to go right down to zero, but it does it doesn't increase linearly any longer. One important area of the curve is this potential for overcompensation. And what this means is that sometimes insect feeding on plants can cause the plant to respond to insect feeding by overcompensating and producing more yield than it ever would if it had no insect pressure at all. So keep this kind of in mind, the concept of tolerance and compensation, as well as overcompensation, when we look at this set of graphs. This is again derived from the same student in our lab who looked at fertility levels in, again, potted plants in a growth chamber that ranged from 0 fertility added to the recommended rate to way above the recommended rate. And what he was measuring here was the mean root mass. And a fascinating set of observations resulted from this work, that as we add insects to these plants, so this was a control plant with no insect larvae on it versus a plant that had 10 larvae on it per plant. As a result of just having diamondback moth larvae on it, the plant responded. And this is canola, by the way. The canola plants responded by producing greater leaf mass. So now the plants are increasing the amount of area that the roots can cover below ground so that they can better sequester available nutrients and better compensate for diamondback moth attack. So the message then is not to cut back on fertility applications because diamondback moth tend to lay most eggs or survive better at optimal levels of fertility, but even though that's the case, these plants can better compensate for herbivory and you end up with a vigorous plant stand. Diamondback moth don't treat all host plants similarly. Here I'd like to draw your attention to some work with 3 different varieties of the very same species, Brassica napus. And we had quite substantial differences when we compared the susceptibility of each of those host plants varieties to diamondback moth egg laying. Looking now beyond the variety aspect to different species of plants, this is brown mustard, Polish canola, cabbage, and Sinapis alba. One clear result from our studies was that white mustard, or Sinapis alba, is most susceptible to diamondback moth egg laying. And most eggs were laid on this species. Brassica rapa, or Polish canola, is also very susceptible to this insect. And Brassica gensea is rather intermediate between Polish canola and between Brassica napus. One interesting result that I'm not presenting here is we also compared in some studies Sinapis arvensis, which is wild mustard. And wild mustard is right up there with Sinapis alba. It's very preferred as a diamondback moth egg-laying site. Diamondback moth larvae grow very rapidly on it and do extremely well on that species. So I think it's very important that controlling diamondback in canola helps by getting rid of wild mustard in crops because anything else is a less suitable host or less preferred host. These are some effects of diamondback moth feeding, again using root mass and how the plants compensate for diamondback moth herbivory. And again, this is the relationship with Brassica napus, much more root mass produced when they're under feeding pressure by diamondback. The same kind of relationship, even stronger with Brassica rapa and Sinapis alba, but with Brassica gensea, it simply did not respond by producing greater root mass as a result of diamondback moth feeding. Well, one very important aspect of this presentation is how to monitor and control these insects. There are a number of ways. Firstly, I think monitoring in crops ought to begin with monitoring for adults. These are the individuals that arrive on airflow from the United States or northern Mexico early in the season, and we ought to have a good idea early in the season whether there's a threat of diamondback moth attack on those crops. Diamondback moth monitoring is best performed for adults with the pheromone trap. A pheromone is a chemical compound given off by females, given off really within very soon after emerging on the first night after these insects emerge. The female will begin calling, which involves moving her abdomen, secreting the pheromone so that it becomes volatile. It's picked up by air currents and carried downwind so that males can follow the odor trail back to the female and mate with her. Remember, they mate at night, so they can't necessarily see each other. So we have been able, or chemical ecologists, I should say, have been able to synthetically prepare pheromone and use it as a monitoring tool by trapping adults. And it can give an indication of the abundance of moths in a given field. This is the standard pheromone monitoring trap for diamondback moth, and it's assisted by having a central portion which contains a rubber piece that has pheromone impregnated on it, and it is attractive to males and used for monitoring. By taking the trap open and counting the number of males, it gives an indication of how many moths are in a given area. This pheromone monitoring trap is operated by a number of agrologists as well as by various specialists within government departments, but it can also be handled very easily by individual farmers. The traps are available to anybody, and they're relatively cheap, they're easy to set up and easy to monitor. Here are some general guidelines for monitoring adults. We've just had a student finish her master's thesis on this very topic, and some of her recommendations are that for best results, hang the traps about 30 to 50 centimeters above the ground. So these insects tend to fly very low, and therefore the best captures are made by hanging the traps only 50 centimeters above the soil surface. Stick them out in the fields early in the season in May. It's easiest to place them along the edges of canola fields for easier access. And finally, the rubber lures only have to be changed once every 6 weeks. We tried changing them more frequently in this research, but it didn't make any difference. Then high numbers of adults in the trap, as many as 100 per week per trap during bloom to early pod, will certainly give an indication that larval numbers are likely to be significant and likely to be very high in those fields. And monitoring of adults then ought to lead to monitoring of larvae, especially if adults are being picked up in the traps. The traps are available. There's a Canadian supply company, Contec, in Victoria, BC, that provides the trap. The approximate cost is only $10 for the trap and the lures. Easy to get a hold of and very easy to monitor, and I'd recommend it personally. A second strategy for monitoring crops is for larvae. Larvae can be monitored by doing whole plant collections or by doing sweep net sampling. Sweep net sampling gives an indication that diamondback moth are present in a given field. This is the contents of my sweep net in southern Alberta, mostly filled with diamondback moth, with a few other stragglers of lygus bug and lady beetles and so on. But this doesn't give an accurate assessment of population density. It gives an indication that diamondback numbers seem to be high, but in order to get a better handle on population density, it's much better to conduct whole plant sampling, and that's recommended by agricultural entomologists. The way that this is done is to remove the plants in an area of about 12 inches square, a tenth of a square meter. Then, so cut off those plants and beat them onto a clean surface and count the number of larvae dislodged from the plants. It's often easiest to take at least I find, a plastic garbage bag into the field, cut off all the plants, gently place them into the bag, and take the bag out of the field in order to bang off the larvae. And often that's only what people do, is by just knocking the plants on a hard surface and seeing what larvae come off. But I really think you should also look through the plant and check it over more carefully. You can see, for example, if there are first instars mining the leaves, if there are pupae attached, that won't come off, or if there are larvae hanging on and still not detaching. To obtain an accurate count, we recommend repeating that procedure in about 5 locations per field. The economic threshold that we use for Diamondback in canola at the pod stage is 200 to 300 larvae per square meter. That translates to about 20 to 30 per square foot, or 2 to 3 larvae per plant. We consider this to be the economic threshold at the pod stage of development, which is the stage that we recommend spraying if spraying is needed. There are 4 products registered for control of Diamondback moth: Lorsban, Matador, Desis, and Malathion. But one very important consideration in applying insecticide is to be concerned about the pre-harvest interval. The pre-harvest interval is the minimum number of days between the last pesticide application and crop cutting or swathing. So it's not the number of days between spraying and combining if you swath in between. It's the days between spraying and swathing. It's very important to follow pre-harvest intervals. The pesticide that is applied to control Diamondback will remain on the plants, and there will be some residues initially, but these degrade fairly rapidly, and if the pre-harvest intervals are followed, the levels of residue on the crop will be at levels below tolerances that have been established by Health Canada. And here are the pre-harvest intervals for the 4 products that are registered. The pre-harvest interval or pre-cutting interval for Lorsban is 21 days, and much less for Matador, Desis, and Malathion. So in summary then, the best management strategy for obtaining profitable crops and preventing losses from Diamondback is to do early monitoring and to limit the use of insecticides to only those fields that reach economic thresholds. I'd like to conclude with what I think is actually about the most important section beyond monitoring and control, or right after monitoring and control, and that is the role that natural enemies can play, because this can affect whether it's necessary to apply insecticide to crops for Diamondback moth in Alberta or Saskatchewan or Manitoba. We've done a number of studies to show that there are 3 different wasp species that can attack Diamondback. These are the 3 species, and these are parasitoids. And just to let you know what I mean by a parasitoid, it's an insect that attacks a host and it eventually kills it. And the way it does it is the parasitoid lays an egg on the body of the host or in the body of the host, and that egg hatches to a larva of the parasitoid that feeds on the host and eventually kills it. We've done a number of studies. This is one set of data from Saskatchewan in the early 90s to show parasitism levels in populations of Diamondback. In this particular survey, we found that about 1/3 of the population was not parasitized and about 67% of the population was. Most of those were Diadegma insulaire, a larval parasitoid that is of crucial importance for controlling Diamondback moth. This is a photograph of Diadegma insulaire, a female next to a Diamondback moth larva. The female wasp has a sharp pointed ovipositor at the tip of her abdomen, and she uses that to stab into the host larva and insert her egg that hatches into a wasp larva inside of its Diamondback moth host. Diadegma is extremely efficient. It's attracted to the odor and the feces given off by Diamondback moth, and it's even attracted to canola leaves that are damaged by feeding of diamondback moth. This diamondback moth larva in my photograph was feeding on this canola flower when a diadigma wasp landed on top. The diamondback moth larva sensed the diadigma, and it tries to escape by hanging on the silken thread below the plant. But the diadigma just sat there. It flew away by the time I took this photograph, but I've witnessed this quite a number of times where the diamondback will try to escape, hang on a silken thread, and eventually it has to move back up to the host plant. And there the diamondback is waiting for it patiently, and it'll stab it with its ovipositor and insert its own egg. The diadigma is unable to parasitize it when it's hanging midair. It needs to anchor its feet on some sort of solid surface. So they're very highly specialized to attack these insects and extremely efficient at natural control. They don't overwinter, as far as we know, in Western Canada, and they're believed to migrate northwards along with diamondback moth. But they can have enormous effects, and I believe very strongly that this insect has saved Alberta farmers millions of dollars. I'd like to draw your attention to 4 outbreaks of diamondback moth since I've worked in Alberta. We had 2 huge outbreaks. In 1995 and 2001, we had vast numbers of hectares that were sprayed, and at a very considerable cost to canola producers. In 2003 and 2005, we also had diamondback moth outbreaks, but we had much less area that was sprayed at a much lower cost to producers, according to our best estimates. From having sampled many canola crops in all 4 of these years, I believe from that sampling that these 2 outbreaks in 2003 and 2005 were every bit as serious as these early ones, but the difference was the presence of Diadigma insulaire. In these outbreaks, Diadigma arrived in large numbers at about the same time that the host insects arrived on trajectory winds from southern North America. They were there, they parasitized their hosts, and they helped really knock down populations. I especially remember the 2005 outbreak where we could tell right away that Diadigma numbers were substantial. We were advising farmers, hold off spraying until the pod stage. Diadigma may solve your problem for you. And by the time the crop was in pod, it was very hard to find diamondback, but Diadigma were everywhere. They had emerged by the millions throughout fields and were attacking and parasitizing any diamondback that happened to be left. Well, how can you tell if you've got Diadigma in your fields? One easy way to tell is by looking at the pupal stage. As I mentioned earlier, the diamondback moth pupa has this lacy covering on the outside of its pupa, whereas the pupa of Diadigma look very different. And the reason for this is that a diamondback moth can be parasitized by a Diadigma, but it can still construct a diamondback moth pupal case. Then, about a day or 2 after the diamondback moth pupates, the Diadigma emerges, having killed the pupa of the diamondback. It emerges and constructs its own pupa. So here's the pupa of Diadigma within the lacy outer covering of diamondback. These are very easy to tell in fields. We're now doing a survey throughout southern and central Alberta to determine the incidence of Diadigma, and hopefully those results should be available in a week or 2 and help guide whether or not we should be advising farmers to spray or hold off. Diadigma can have very beneficial effects. This graph shows some results of research with diamondback moth and feeding that they performed when they weren't parasitized compared to when they were parasitized. And regardless of the fertility level of the plant, the larvae still ate more leaf material when they weren't parasitized compared to when they were parasitized. So they not only kill their hosts, but they also cut down on the amount of feeding that those individuals are doing. In terms of survival, the parasitoid performs a little differently than diamondback. It seems to prefer diamondback moth larvae that were reared on the most fertile plants, but still performed well under plants that had recommended levels of fertility, and performed most poorly in terms of survival levels on diamondback grown on plants that received no fertility. One fascinating aspect of the ecology of canola is shown in this slide. In 2005, we were interested in making some collections of Diadigma pupae to bring back so that we could rear out Diadigma that I could have for teaching in my courses. And we found an interesting phenomenon. In several of the cases, we reared out Diadigma as we expected. This is a female with her ovipositor. But in some cases, we never got Diadigma at all. We got these tiny little wasps that were a species of Catalochus. When we investigated further, these are actually parasitoids of Diadigma. So the Catalochus is a parasitoid of a parasitoid, or we call it a hyperparasitoid. I guess in the sense of farming, these are detrimental because they kill off the good guys, but they're not very abundant. And to me, they just speak of the wonderful diversity that we have in some of our agricultural cropping systems. But there are other natural enemies besides Diadigma that are worth noting, and I think it's wise that farmers and agronomists consider populations of lady beetles, because they consume large numbers of eggs, large numbers of diamondback moth larvae, and they're an important biological control agent. Lacewing larvae are also important predators in canola crops. They attack their prey by seizing them with their large sucking jaws. They inject a paralyzing venom into their host insects and then draw the material out of the body of their hosts. They feed on aphids in the crop, they feed on insect eggs and insect larvae, like diamondback moth and like small Bertha armyworm larvae. And one important predator that many farmers might not appreciate, or agrologists, are the carabid beetles. And the reason they might go unnoticed is because they're active primarily at night. They can be incredibly abundant. My students in only an acre or so of cropland of canola in Lacombe and in Edmonton have found 50 different species of these beetles that are patrolling the crop canopy in canola, hunting for any prey, and in this case for any diamondback larvae that may have occurred. One of the other aspects is that we can have fungal pathogens that can help attack diamondback. For example, this pathogen that has killed this diamondback moth larva is Zoethra radicans, and they can be responsible for massive reductions in diamondback populations. For example, in the outbreak of 1995, throughout much of central Saskatchewan, over a one-day period, I think there was about 1.5 inches of rain that occurred during the peak of diamondback moth spraying. That was followed by 2 days of extremely humid conditions, which allowed an epizootic of Zoethra to develop, which more or less decimated the populations, and spraying was really no longer necessary. One other, this is just a slide of diamondback that are attacked by fungal pathogens. And one other consideration that I think farmers ought to take into account this year are the rising lygus bug populations. We're advising that the economic thresholds for diamondback are for pod stage canola, that leaves can take a considerable amount of defoliation before it's necessary to spray, but also it's harder to kill diamondback and lygus, which can be present in the crop at the same time, if there's a canopy of leaves that prevent carry of the insecticide through the crop canopy. So once the crop is senesced, diamondback moth larvae will still be there, and lygus bug numbers will also, if they are present, can be killed by the insecticide as well. Again, here's a photograph of a diseased larva of diamondback. Our models show that this insect is only going to get worse. We've done some projections of climate change. Under current climatic conditions, most of southern prairies can have about 4 generations per year of diamondback, with only a climate change increase of 2 degrees, which is much lower than predictions. This is increasing the number of generations through much of the southern prairies to 5 instead of 4. So our thinking is that this insect is going to be around for a long time yet. So in summary then, diamondback moth can complete as many as 4 generations per year in the southern prairies in the summer. Most damage is inflicted by the final generations when crops are in the pod stage of development. Insecticide applications are recommended for populations at 2 to 300 per square meter, or 20 to 30 per square foot, or 2 to 3 per plant. And finally, it's recommended that canola growers apply insecticide only later in the season if possible. In the past, the parasitoids and predators have sometimes eliminated their populations, and insecticide sprays also kill those beneficial insects. So Rick, that concludes my information on diamondback moth.
Thank you, Lloyd. That was excellent. We do have some questions coming in, and anyone interested in asking a question, you can type that into your little control panel. If it's closed down on you, just click on that orange arrow in the top right-hand corner of your screen. First question, Lloyd, comes from Jonathan. Is there a ratio or method to determine the number of females in an area if one is capturing only male moths?
Jonathan, that correlation hasn't been done precisely. We're interested in it, but in general, we assume that it's a fair guess that male and female numbers are about the same. I know you're thinking that the pheromone traps are picking up just males, but what about the females? But in general, when we've sampled fields, the ratio has pretty much been 50-50. But I agree with you. I think we need to have a look at that maybe a little more closely.
Okay, next question comes from Uwe. Which of the 3 species of wasp is more abundant in central Alberta?
Very clearly, it is Diadema insulaire. I have seen Microplytus platellae and Diadromus throughout the prairies, but never as abundantly as Diadema insulaire, which is our principal parasitoid.
Okay, question from Aaron. What effect do environmental conditions have on diamondback life cycles? Example, like extremely wet/dry conditions we had this year.
Aaron, extremely wet conditions have been linked to controlling diamondback, drowning small larvae, especially the tiny second instars that come through the leaves and begin feeding above the surface. So heavy rainfall has been associated with drowning out some of those larvae. I think my hunch is that in some areas of the province this helped diamondback infestations from being more severe than they really are. In some areas where there's irrigation, irrigation has been recommended as a natural cultural strategy for diamondback moth control. But it's a pretty resilient insect, Aaron. Remember, the first instars are protected within the leaves, so they can hunker down as well and survive heavy rain.
Okay, next question is from Jennifer. How long does it take diadegma to kill the diamondback host?
I have that information. I'm going by my memory banks now. We've done a number of lab studies on this. It's usually been about, depending on temperature again, about 2 weeks. So they prefer to parasitize second instar larvae. They will parasitize first, second, third, and fourth. They prefer second or third instars. And then it takes 2 to 3 weeks till the host is dead. But there's less feeding, and they can still really keep the population from exploding if they arrive in high numbers.
Okay, a follow-up from Uwe. Which of the 3 insecticides is less harmful on predators?
We have no information on that. In general, organophosphates like chlorpyrifos or lorsban have been linked with greater non-target damage, but really I'm not aware of any research that's conclusively shown that in canola in Western Canada.
Okay, question from Dennis. This one deals with lygus, but are there any natural predators of lygus bugs?
Dennis, we have natural parasitoids of lygus. They're a parasitic wasp in a different family than the diadegma wasp. They're in the genus Parastinus, and we have several species of them throughout Western Canada that parasitize lygus bugs in crops. As far as predators go, not too many to my knowledge. The eggs are laid right inside of canola stems, so they're protected there from predators. There is an egg parasitoid that we found that can get the eggs. And the larvae are pretty fast moving. Sometimes they can be taken by lacewing larvae, but I've watched them in the crop canopy quite often, and they're fairly fast moving. So I would say parasitoids are more important for that insect than predators.
Thanks, Lloyd. Question from Yulan. On your slide of impact of fertilizer on root mass weight, can you repeat the conclusion? It seems when too much fertilizer, the root mass drops.
The root mass increase, Yulan, was not as great as I recall under high levels of fertility, but no, the root mass did not drop. It just didn't increase as much. I'm just looking at the graph now, and say at the recommended rate of fertility, we had more than double the amount of root mass with herbivory. It didn't double at the very high rate, but it still increased significantly beyond what it was for plants that hadn't have any diamondback on them at all.
Okay, next question comes from Keith, looking for confirmation from you, Lloyd, that 5 to 10 diamondback moth larvae in 10 sweeps is relatively low.
Keith, I can't confirm that because we don't know how much, we have no quantitative data on how efficiently sweep net captures are for getting diamondback in the field. So 5 to, did you say, what, could you repeat that?
The question was 5 to 10 larvae in 10 sweeps.
I would say it seems low, but no, I won't confirm that because we really haven't quantified what % of the population we're picking up with sweep nets.
And a follow-up there, which of the insecticides is less toxic to honeybees?
It seems, to my knowledge, that there is some evidence that the synthetic pyrethroids, that Desis and Matador, have some repellent effect on beneficial insects like honeybees. So they tend to avoid areas that were sprayed with those insecticides more so than the other organophosphates.
Okay, next question comes from Michael. Lloyd, any relationship between treatment early in season for cabbage seed pod weevil and later in season infestation levels of diamondback moth?
We haven't done any correlations between those two pests, but suffice to say that a spray for cabbage seed pod weevil, if it's done according to recommendation, which is at 10% flower, from what I saw this year, diamondback moth numbers throughout southern Alberta where the weevil is found, diamondbacks were not in the less vulnerable pupal stage or the first instar stage. We were getting second and third instar larvae at that time. So a spray for weevils ought to have substantially reduced diamondback moth populations in those fields as well.
Here's a follow-up just on that sweeping question about 5 to 10 in the sweeps. Keith says they're finding 1 larvae per 12 plants with that same sweeping pattern.
Well, I think, Keith, you've verified, I guess, in your study that that's a low number to be able to get in sweeps, and that low a number of individuals per plant is substantially below the economic threshold. So it very much seems like it relates to a low number between sweeps and individual plant counts.
And another question from Jonathan going back to the root mass. Did this root mass response occur early in the canola growth stage, later, or at all stages? And was there a direct correlation to yield increase? We
never followed those plants. Unfortunately, it's one of our greatest regrets. We never followed those plants to yield. But we are going to be working this winter to redo that and get the yield data. But having said that, the larvae were placed on the plants when they were in the rosette stage of development. So the root mass increase began relatively early. And the thinking there was to inoculate the plants with larvae at about the time that diamondback moth would be attacking. So I believe that the root mass increases ought to have occurred soon enough that they would have caused an overcompensation in terms of yield.
Okay, and a question from Tracy. For the first instar larvae, is it true they struggle dealing with daily rains? Does it inhibit their feeding activities?
Well, first instars in canola are feeding within the leaves, between the upper and the lower epidermis. So I don't believe that rainfall has really any effect on them. I suppose it's possible that water and so on could drown them inside their feeding tunnels, but I doubt that very much. I think they're very protected. I think it's the second instars that are still very small, that emerge from the leaves, that can be susceptible to getting washed away and drowned by rain at that stage.
Okay. Thanks, Lloyd. That appears to be the last of our questions. So again, on behalf of the Alberta canola producers and everybody online, thank you very much, Lloyd. I think everyone would agree with me that this was a very valuable and timely presentation. Just a reminder to everyone online, there will be a short survey following when you log off. We'd certainly appreciate if you do it. Visit our website for any more information that you're looking for about our webinars, be it recorded or upcoming ones. And if you have any ideas or suggestions, please feel free to drop me an email or give me a call. So once again, on behalf of the Alberta Canola Producers Commission and the canola growers within Alberta, thank you, Lloyd, and thank you to everyone for attending today's webinar, and have a safe and happy end to the summer season. Goodbye, everyone.